There are three pharmaceutical companies that make up the United States' $8 billion insulin market: Eli Lilly, Novo Nordisk, and Sanofi. These big three companies make up more than 90% of the $20 billion of insulin revenue worldwide, with only 7 or so companies making up the remaining percentage. But that might not be what you'd expect for a drug discovered over 100 years ago. Instead of a bunch of competing companies making cheap generics, there's a global oligopoly. And part of that is due to the fact that the insulin available today is different from the insulin of the early 20th century, thankfully. But those improvements come with a price. So in today's video, I'll tell you the story of how insulin was brought to market, how pharmaceutical companies in the 1920s set the stage for insulin production today, and how those companies turned insulin from a crude pancreatic extract into a dependable, life-saving product. Let me start by introducing you to our first company, Eli Lilly and Company. When Eli Lilly started the company in 1876, it was considered an ethical drug maker, a term that meant they adhered to the ethical standards of the American Medical Association. These companies didn't advertise much, they didn't hold patents, and they didn't introduce new drugs. No gimmicks, no secret recipes, just good, honest drugs. But by the end of the 19th century, they were losing market share to European chemical companies who were rolling out new and effective drugs like aspirin. And this was by design. These companies hired chemists and pharmacologists with the explicit intention of creating marketable drugs. So the ethical drug makers like Lilly would have to change something if they wanted to stay afloat. And it was really one strategic thinker at Lilly that turned things around: Eli Lilly Jr., the founder's grandson. Eli Jr. went to pharmacy school, but was hired as the sole member of Lilly's economic department in 1907. And to give you an idea of the kind of guy we're dealing with here, at one point, he invented a machine that made filling bottles more efficient, saving the company $7,500 a year in spillage. Then he started lining their wooden barrels with copper because he calculated that the alcohol seeping into the barrels was costing them money. Big business guy. In 1909, his father, J.K. Lilly, promoted him to the manufacturing division. And by all accounts, Eli Jr. became even more obsessed with the economics of the company. Within a few years, he'd created a new factory building dedicated entirely to producing gelatin capsules. And by World War I, Lilly could make 2.5 million capsules a day, and even exported their products overseas. Eli Lilly was still considered an ethical pharma company, just one with incredibly efficient production. But if Lilly wanted to compete with the European companies, they'd need to bring new drugs to market. Step one was reworking their science department into a department that would find or develop new and profitable drugs. And that meant they'd need a super unique hire, someone who knew both business and medicine. So in 1919, they hired and later promoted Dr. George Clues as the new director of research. Clues had a doctorate in chemistry, but was academically curious about everything, which made him a great fit for the job. And for the most part, Lilly just kind of let Clues research whatever he wanted to on behalf of the company. Even if it was exploratory research, the hope was that it would pay off in the long run in the form of new medicines or relationships with researchers. Now, Clues eventually decided to focus on diabetes medications, in part because there was just nothing out there. If you remember from the last video, the best treatments for diabetes at this point were essentially starvation diets. So, Dr. Clues went on the hunt for any medicine that might treat diabetes. And one day, in 1921, he was talking to a University of Toronto alumni who told him about some interesting research happening at his alma mater. Clues heard the researchers were going to be at a conference in New Haven, Connecticut in December, so he was like, I'll check them out. And this is where he meets our crew from the last video. 3 researchers from the University of Toronto: J.J.R. McLeod, Frederick Banting, and his student, Charles Best. They were at the conference presenting their results using this brown mush that they had extracted from dog pancreases. They were tentatively calling this extract iletin, but an earlier scientist had named the active component insulin. The team from U of T didn't have a product to sell. They hadn't even done human experiments yet. But their animal experiments were enough to get Clues interested. So after the conference, he contacted McLeod, the head of the Toronto Research Group, and proposed a partnership between Lilly and U of T. And McLeod said no. For now. See, the University of Toronto had a unique relationship with a lab called Connaught Antitoxin Laboratory. In their own words, they were, quote, established for research investigation in preventive medicine and for the production and distribution of all public health biological products at minimum prices. And they had a unique relationship to U of T. Like, the lab's facilities were in the same medical building as McLeod's physiology department, so Connaught would be the logical producer for the extract. This is also around the time that a biochemist named James Collip comes into play. While originally from the University of Alberta at Edmonton, he had been awarded a fellowship opportunity which allowed him to do a sabbatical at the U of T. And at the end of 1921 into 1922, Collip figured out a process for refining the pancreatic extract and improving the purity of the final product. This was some of the first clinically usable insulin and led to their first successful human trials. So now they've got something to produce. And on January 25, 1922, the Toronto researchers and Connaught entered into an agreement. Connaught would provide the facilities, some funding, and resources for insulin production, and in return, the researchers wouldn't work with a pharmaceutical company for a certain amount of time, and they wouldn't make changes to research policy unless they all talked about it first. And there were some interpersonal reasons for that last part. The Toronto researchers weren't exactly getting along at this point, so the agreement also served as a way to keep everyone on the same team. And for a little while, it worked out well. The lab helped make enough of the extract to supply more clinical trials, as well as for some Canadian doctors and public health organizations to use clinically. The Toronto team had conducted their first successful human trials at Toronto General Hospital in early 1922, and then more doctors treated a few other patients over the next few months. But it was clear that they'd need help if they wanted to scale up production even further. Unfortunately, there was one big snag. Collip hadn't properly documented his process for making insulin, and he couldn't figure out how to reproduce his work. So unfortunately, in February of 1922, just as the hype for insulin was building, the University of Toronto and Connaught entered into an insulin shortage. It must have been devastating. At this point, there were already patients who depended on insulin. Like, people's lives had been saved by this miracle of modern medicine, but then all of a sudden, there wasn't enough to go around. Meanwhile, Clues at Eli Lilly is still calling and sending letters asking to work together, to which McLeod said, "We're still good, we'll let you know though." But in reality, the Toronto group was in a tricky spot. They needed more insulin to correct the shortage, and McLeod especially was scared that some other lab would come in, patent insulin, and prevent others from making it. So in April of 1922, they decided to apply for a patent, which is something that folks in medicine and academia didn't really do back then. I go into detail about why in this video here, but the Toronto team decided to do it for two big reasons. The first was safety. Neither the Canadian nor the United States governments regulated drugs like insulin back then. Like, the US FDA didn't make drug companies show that their drugs were safe until 1938. The second big priority was the public trust. If the Toronto crew patented the extract, they could make sure that anyone who wanted a license could get one. It would also prevent a for-profit competitor from taking out the patent and creating a monopoly. Now, Banting was a member of the medical profession, so he didn't want his name on the patent from an ethical perspective. But his collaborators weren't medical doctors, so at first, the original application only listed Best and Collip's names. But eventually, attorneys decided that Banting's name would need to be on it too, since he was one of the discoverers. In the end, the patent was written in such a way that emphasizes what the extract does more than how it was made, which is going to become relevant here in a second when we start talking about process patents. When the patent application went through, Collip, Best, and Banting assigned their intellectual property to U of T as a public trust, meaning it would benefit the public. They technically sold their rights for the symbolic cost of $1 apiece. Frederick Banting is often quoted as saying, "Insulin does not belong to me. It belongs to the world." This patent process lasted until the end of 1922 and was completed in 1923. This fish thing is on there because McLeod figured out a way of extracting insulin from fish pancreas during that time, so might as well throw it in there. By mid-May, they'd figured out how to make insulin again, but only in tiny amounts. It would be enough for more clinical testing, but not much else. Meanwhile, the demand for the extract was blowing up, because in early May, McLeod presented a paper at the meeting of the Association of American Physicians. This is where he debuts the name insulin, from the Latin insula, meaning island, referring to the islets of Langerhans in the pancreas where insulin is made. After this meeting, demand exceeds their already limited supply. It got to the point where doctors started showing up in Toronto to beg for insulin for their patients. And this is when McLeod realized that they would need more help with production. Connaught Labs might have been able to supply Canada, but not the world. They tried looking for partners in the US that would agree to handle the insulin patent on the same terms as they did at U of T, but they couldn't find a great match. So, the University of Toronto decided that they'd take responsibility for insulin manufacture in Canada and the US. But they should probably take that meeting with Clues and Eli Lilly. So, going into this meeting, it was going to be a super unique partnership, and it's all happening while the Toronto team is working on the patent application too. The university would handle the rights to the patent in both Canada and the United States, and by September of 22, the university's board of governors had put together a special insulin committee. The committee would administer the patent licenses to different companies and collect a small royalty fee. Between 1923 and 1967, they made a total of 8 million Canadian dollars in royalties. And part of that money went towards the costs incurred from testing the insulin made by the licensees. They meant it when they said they wanted to make sure the insulin was up to standard. For the first year, U of T would grant an exclusive license to Eli Lilly for insulin production and sale in the Americas, except for Canada. They could have given licenses to multiple companies, but giving it to a single company was just more efficient. They probably knew that this decision to work with Lilly would be criticized, but their number one priority was producing more insulin and getting it to patients quickly. According to a letter from the researchers to the president of the university, quote, "We have chosen to collaborate with Eli Lilly and Company. We recognize this firm will be placed at an advantage over its competitors through this collaboration with us, but we believe it is much less serious than there should be further delay in proceeding intensively with production on a large scale. And moreover, we propose to give other firms, as well as hospitals and other non-commercial concerns, every chance to do the best they can by publishing the details of the method as at present used by us in the Connaught laboratories in full at an early date, within 3 months. By this step, our proposed cooperation with the Lilly company cannot be criticized as unethical or unfair or as in any way prejudicial to the free manufacture of insulin." The researchers still planned on publishing their production methods so that other drug makers could hit the ground running once Lilly's exclusivity period was up. And during that first year, Lilly and Toronto promised that they would share all their insulin research with each other, and that Lilly would provide cheap insulin to certain physicians who treated certain cases, like diabetes in kids. And after that year, Lilly would license insulin from U of T just like any other licensee would. They signed the agreement at the end of May, and their year-long exclusivity period would run from June 28, 1922 to July 1st, 1923. Now, Clues was thrilled with this because even that short exclusivity window was a huge head start. Being a category leader comes with disproportionately large marketing advantages. And speaking of marketing, Eli Lilly would need a brand name. After all, this was going to be Americans' first exposure to commercially available insulin. And so at first, they went with the brand name Ileton, but were required to include the clarifying generic name Insulin Lilly. That way, when other insulins hit the market, it would be clear that they had the same active ingredient. Regardless of the name, they now had a first-in-class drug with a decent-sized market, about 25,000 type 1 diabetic patients in the US by October of 1923, and these folks would need to take the drug every day for the rest of their lives. Alright, so at this point in the story, Lilly and Toronto have struck a deal. Now the challenge is getting insulin to the rest of the world. While McLeod was working on the details of the deal, Clues went back to the Lilly headquarters in Indiana to gather as many livestock pancreases as possible. For my non-American viewers, Indiana is in a part of the country with tons of agriculture and farming, so they were in a great spot. Also, how fun is this map, huh? Now, before they could properly start production, both the university and Eli Lilly had some more research to do. Because while they were able to recreate Collip's methods, they weren't able to get that much usable insulin out of a given pound of pancreas. The researchers at Toronto and Connaught had some luck using acetone, but doctors complained that the resulting insulin wasn't super potent. And that was a common theme for the first few batches. Increased yields came at the cost of potency. Luckily, Eli Lilly's lead chemist, George Walden, made a huge breakthrough in the fall of 1922. It was called isoelectric precipitation. Here's how it works. Any given proteins in a solution, like insulin, have a net charge to them. But at a specific acidity, you can get the net charge of those proteins to be zero, or isoelectric. When that happens, the proteins clump together, or precipitate, out of the solution. In this case, yielding more usable insulin. From Lilly's perspective, the new process meant that it would be cheaper to make insulin in bulk. And from a clinician's perspective, it meant the dosage would be more standardized. Now, Lilly wanted to patent the isoelectric process for insulin, but some members of the insulin committee thought that the patent's wording was a little too broad, which might have led to an American monopoly on insulin if it were approved. At the same time, a separate American researcher named Philip Schaffer, who's mentioned here, had recently discovered the same thing. So Lilly probably wouldn't have been able to get the patent. Either way, the whole process led to a new patent pooling situation where anyone who licensed insulin from the insulin committee would have to share new insulin patents with the U of T, then the committee would license them out. It was very cooperative science. By the end of their first year, Clues proudly claimed that Lilly was finally able to meet the North American demand for insulin. But in the middle of all this tinkering with the extract, insulin was still an injectable medicine. And considering how folks with diabetes needed to take insulin multiple times a day, having a doctor do each shot wouldn't be sustainable. So Lilly started shipping reusable syringes that patients would learn to operate. And that first year of sales was incredible for Lilly. They made over $1 million in 1924 off of just insulin. And by 1926, they claimed to be the third biggest pharmaceutical maker in the world. So by the time that other companies could finally license insulin in late 1923, Lilly had two big advantages. The first was branding. They were the first mover in the category and had built trust with their customers. And the second was economies of scale. They had the resources to keep production costs low, which allowed them to sell insulin at a low price, which disincentivized competitors from entering the market. But while Lilly got exclusive rights in the US for a year, the patent for insulin in the United Kingdom went to the UK's Medical Research Council, or MRC. The MRC bought a few shipments of Lilly insulin to cover them in the short term, but eventually the MRC contracted a few British pharma companies like Burroughs Wellcome to produce the drug. And at first, British insulin was more expensive than American-made insulin because the UK hadn't achieved economies of scale like Lilly had. 100 units of the first British-made insulin sold for 25 shillings in April of 1923, or about £76 in 2023 currency. But by the summer of 1923, the MRC had figured out production enough to supply Britain. And by the following summer, Burroughs Wellcome started selling their insulin in Mexico and Latin America. And of course, Britain and North America weren't the only insulin manufacturers. I butcher their name every time, but the German company Hoechst AG started selling insulin in October of 1923. But since the German economy still wasn't doing that well after World War I, they didn't become a huge insulin producer right away. There was one more important manufacturer though. I should say two. You'll see what I mean. This is August Krogh, the Nobel Prize winner for medicine in 1920. His wife, Marie, was a doctor herself and actually the first woman to earn a doctoral degree in medicine in Denmark. And in 1922, she was diagnosed with diabetes. In fall of that year, the Kroghs went on a post-Nobel lecture tour of North America where they kept hearing about the insulin research in Toronto. So August arranged a meeting with Banting and MacLeod and spent a few days seeing diabetes patients, checking out insulin production, and attending a meeting of diabetes specialists. But most importantly, Krogh left America with the University of Toronto's blessing to make and sell insulin in Scandinavia. Now, at the time, you couldn't actually patent drugs in Scandinavian countries, so this was more of an endorsement than an official license. When the Kroghs got back to Copenhagen, August teamed up with a doctor named Hans Christian Hagedorn and assembled a team that began testing insulin immediately. And after some success, they partnered with a local pharmaceutical company called, in English, Lion Chemical Factory. I am not even going to attempt the Danish pronunciation, y'all have heard my German. They spent early 1923 extracting insulin from fish and pig pancreases, improving potency, and figuring out that you could improve the yield of insulin if you froze the pancreas immediately after cutting it out of the animal. And by June, they released their version of insulin under the brand name Insulin Leo. The name was part of a deal they'd made with Lion Chemical Factory in return for covering some of the startup costs. So production went well, clinical testing went well, and after paying back Lion, Krogh and Hagedorn decided to officially start their own factory, the Nordisk Insulin Laboratorium. But after a few months, two disgruntled Nordisk employees, Thorvald and Harold Peterson, left Nordisk to start their own competing insulin company. And they called this new company Novo, and officially launched their Insulin Novo in 1925. Novo's insulin debuted at half the price of Nordisk insulin, which of course meant that patients saved money, but it also set the tone for a decades-long competition between the two Danish companies. Novo and Nordisk would eventually merge into one company, Novo Nordisk. Yep, that Novo Nordisk. But for most of the 20th century, they were rivals. By the 1930s, the global insulin market was Eli Lilly, Nordisk, Novo, and everyone else. But in reality, it was Eli Lilly's game to lose. They had such a lead over the other brands that they remained the #1 insulin seller in the United States until 2006. So before we go on, let's talk about prices. In 1923, when Lilly still had its exclusivity agreement, a daily course of insulin cost anywhere from 40 cents to $1 American, which is about $7 to $18 a day in 2024 dollars. That's a serious chunk of money. But just 5 years after the introduction of insulin, a global competitive market for the drug had emerged. The intellectual property was readily available, so anyone with access to livestock pancreases could start an insulin company. And while every country and every market was different, the cumulative effect was that the price went down dramatically. Like this article from 1923 shows insulin prices in Canada dropping from 5 cents a unit to 2 cents. And at a time when patients needed 15 to 20 units a day on average, that meant that cost was dropping from about 75 cents to $1 a day to 30 to 40 cents per day. While a full vial of insulin sold for $1 when it first came out, it would cost less than 5 cents in the 1930s. But of course, this price drop couldn't continue forever, since insulin as it was in 1923 wouldn't be on the market for much longer. What people called insulin in the 1920s was a crude and impure pancreatic extract. It was still mostly coming from cow and pig pancreas. Insulin from fish was effective too, but it was much easier to get the pancreas from cows and pigs. And to be fair, purification technology was getting a little better. But that also meant that insulin was absorbed from the bloodstream much faster than before. So patients would experience spiky blood glucose levels and have to inject themselves all the time. It would be great to have insulin that lasted an entire day. In the decade after insulin came to market, a few different labs tried adding things to insulin to get it to last longer in the body. One group tried adding it to a gum arabic mixture, another tried mixing it with lecithin or oils, and some experiments involved trying to delay absorption by adding adrenaline, which didn't really work for them. But then in 1934, the Nordisk team led by Hagedorn tried adding a type of protein called protamines. They were thought to be these small, super simple proteins with really low solubility at the acidity of normal human blood. So the idea was that by adding the two together, insulin would take on the low solubility and thus slow release time of protamine. Now, one of the places you get protamines from is fish sperm. So Nordisk tested a bunch of protamines from different fish and discovered that protamines from rainbow trout sperm were the best. Do with that information what you will. When it came time to test in humans, the resulting protamine insulin was less soluble and dissolved more slowly than the old-style insulin, which used hydrochloric acid. And their experiment showed that you could get away with 2 shots of protamine insulin a day, one in the morning and one before bed. Meanwhile, folks on the old insulin needed 3 to 5 shots a day. Hagedorn published his trial results on protamine insulin in early 1935 and got it approved by Danish authorities in early 1936. And since you still couldn't patent drugs in Denmark, he patented the production process. Next, they took out patents around Europe and started selling licenses to pretty much anyone but Novo. I tell you, it's a real enemies to lovers storyline here. Nordisk started selling protamine insulin in the US in August of 1935 and filed a patent early the next year. So, at this point, Lilly is old news, right? Patients had wanted long-lasting insulin for years, and here comes Nordisk with a breakthrough technology. Well, at first, Clues wrote a letter to Hagedorn and asked him for an exclusive license for protamine insulin in the United States. They'd be happy to pay royalties. But Hagedorn wasn't so sure about working with Lilly. So he went to Charles Best at Toronto and decided that Hagedorn would give the rights to protamine insulin production in the US to the Insulin Committee, who would then license it to American pharma companies and pay Nordisk a 5% royalty that way. It was similar to how the Insulin Committee had dealt with the original insulin patent. But Nordisk wasn't the only one working on long-lasting insulin. 2 Canadian scientists at Knott Labs, Scott and Fisher, had been keeping up with the Danish research. And when protamine insulin came out, they weren't convinced that it was actually longer lasting than traditional insulin. See, they had had some luck slowing down insulin solubility by adding zinc, so they had another idea. Could they slow down absorption even further by adding zinc to protamine insulin? Sure enough, this new compound, zinc protamine insulin, or ZPI, slowed down absorption even more than plain protamine insulin. ZPI had the added benefit of shipping in a single bottle too, as opposed to Nordisk's protamine insulin, which shipped in 2 bottles. One for insulin, and one for protamine in a special buffer solution. The University of Toronto took out a patent for ZPI, started licensing it to other companies, and it quickly became the most popular insulin in North America. Meanwhile, back in Denmark, Novo tried to make their own zinc protamine insulin, but Nordisk sued them because you can't make ZPI without the PI part, which they had the patent for. The lawsuit eventually got kicked up to their Supreme Court, where the judge ruled in favor of Nordisk. But by then, it was the middle of World War II, so Nordisk granted Novo a super cheap production license which would ensure that Scandinavia had plenty of affordable insulin throughout the war. Basically, the whole episode was a wash. Now, while Nordisk took some losses throughout the war, they had mostly recovered by 1949. Novo, on the other hand, were starting to introduce some non-insulin products, so they ended up in a better financial situation after the war. This is also lining up with a major supply and demand issue for insulin in the post-war period. First, the supply. Beef and pork were getting more expensive, which brought up the price of insulin since it was still based on cow and pig pancreas. Second, the incidence of type 1 diabetes was increasing all over the world, which, of course, meant more demand for insulin. It was time for the pharma companies to figure something out. During World War II, two Nordisk scientists figured out how to get protamine insulin to form crystals, which gave the resulting insulin two big benefits. The most important benefit was that it could be taken safely alongside fast-acting insulin, and that meant that diabetic patients could keep their baseline glucose low, then take more insulin as needed after a meal. It hit the Scandinavian market first in 1946, 1947, then they gave the Canadian rights to the Insulin Committee and applied for a patent in the United States, which they got in 1951. Outside of Scandinavia, this version of insulin got the name NPH for neutral protamine hagedorn insulin. And within a few years, this stuff had exploded in popularity. By 1952, 37% of insulin sales in the United States were NPH insulin. Now, Novo knew that they had to step up their game. But how did they come out with something better than NPH without using protamine crystals and thus violating a Nordisk patent again? Their answer was a buffer solution of acetate. The buffer reduced the solubility of the insulin and kept it working for longer, giving them a long-lasting insulin without protamine. This slow insulin became known as lenta insulin, lenta meaning slow, and this led to two other variations. Ultra-lenta, which was less soluble and acted even more slowly than regular lenta insulin, and semi-lenti, which acted more quickly, more like old-school insulin. Novo got patents in dozens of countries and filed for a US patent in 1951, but it wasn't granted until 1959. Importantly, that let them license lenta insulin to Eli Lilly, who was still the biggest insulin maker in the United States. Lilly branded it as lentiilotin in 1954, with the semi- and ultra-lentiilotins following in 1957, although NPH insulin still sold the best. For those keeping score at home, that's a lot of innovation from Denmark, and not a lot from Eli Lilly in like 30 years. And throughout the 60s, Novo came out with even better versions, including Act Rapid, which, as the name suggests, acts quickly, and Rapitard, which starts its effects quickly, but lasts a long time. The 1950s also saw the invention of other drugs for diabetes like metformin. And unlike how insulin clears blood sugar by letting glucose into your cells, metformin keeps your liver from releasing so much glucose to begin with. These drugs were significant too because they were taken by mouth, which made them more accessible than injections. But the biggest challenge going into the 60s was the source material. Aside from rising costs, some patients were getting these local reactions around the injection site, and some developed antibodies to insulin. And antibodies are a good clue that an immune reaction has happened, so scientists wondered what might be causing the reaction to insulin in the first place. An experiment in the late 60s found proteins like proinsulin left over in insulin preparations, and researchers concluded that patients were probably reacting to those, not the insulin itself. So on paper, the next challenge was simple. Make an insulin product without those leftover proteins. So researchers used a new method of chromatography to get the extraneous proteins out of the pancreatic extracts, and it worked. In 1969, a researcher at Novo published his work on the product and filed for a patent. And since this stuff had only one component, the insulin, Novo called it monocomponent, or MC insulin. And in 1970, a clinical trial, conducted in a Novo-sponsored hospital, showed that MC insulin created no or very few antibodies even after a year of treatment. Scientists at Eli Lilly figured out how to separate the non-insulin proteins out of the solution too, and they called it single-peak insulin. The result was that this MC and single-peak insulin were so much more pure than their competitors that other European companies just started buying insulin in bulk from Novo. Nordisk and Germany's Hoechst were some of the only ones who kept producing their own insulin. Canada's Connaught was still around too, although by the 80s, the university had sold them off. They were bought by the French company Rhône-Poulenc by 1989, who then merged with Hoechst in 1999 to become Aventis, which was bought by Sanofi in 2004. And that finally sets the stage for our three players from the intro: Lilly, Sanofi, and Novo Nordisk. By 1980, Eli Lilly had about 60% of the global market for insulin, while Novo and Nordisk, which at this point were still two separate companies, held most of the remaining 40%. Now, even in the early 1980s, insulin was still made using cow and pig pancreases, and pharma companies couldn't totally eliminate allergic reactions in patients. Cow, pig, and human insulin are similar, but not identical. So the next big challenge was creating authentic human insulin. And it took a revolution in genetic engineering, plus a new model of funding to figure out how to commercialize human insulin. In the next video, we'll cover genetically engineered insulin, the birth of biotech, and modern insulin analogs.